Okay. So thank you. It's a great pleasure to be here. My first time in Ireland and in Cork. And very cute city. I had the opportunity to visit the city yesterday. Really beautiful city. So today I would like to make an update on cancer immunotherapy from the bench of the bedside. That's the area I've been working on over the past 25 years, with major progress now that I would like to review with you. So when one thing about the UPMC Hillman Cancer Center, I think Chuck talked about that, I think it's fair to say that it is widely recognized as being the hub of human cancer immunology since its creation in 1985. So at this time, our first founding director was Ron Eberman, who moved from the NIH, was hired by the UPMC leadership, Dr. Dietrich and Dr. Romoff. I think it was very visionary at this time to hire cancer immunologists because there was no really evidence that this was a way to go in the clinic. There was some interesting preclinical data in vitro and in mice, but not in human. And Dr. Eberman made some seminal contribution in the field, discovering a specific subset of cells called NK cells, which play a role in cancer immunology. The first thing that did Dr. Eberman is hire promising and talented oncologist, Dr. Kirkwood, who is now one of the main leaders in melanoma worldwide and immunotherapy, and in particular in the adjuvant setting, and with a team of other investigators, scientists, which are widely known in the field. They developed for years and years approach to stimulate immune response. At this time, we thought that vaccine would make the trick, that we should engineer better vaccine to stimulate immune response, and we managed to stimulate immune response in patients, but this was not enough. We couldn't translate this immune response into clinical benefits for the patient. And this has been the long history of cancer immunology for almost 2 decades. So 2002 was when we moved in this new building, which is our home since 2002, our National Cancer Center, the home of both the research pavilion and the clinic. So for somebody like me who's doing translational research, is very important, the access to sample and having the patient very close. And as I said, what happens, and many over the past 10 years, is finally we have evidence that cancer immunotherapy works in the clinic. Well, I'm going to talk a little bit about that. It's been recognized as being a major breakthrough in the field of oncology. And based on that, I think this has enhanced the effort of UPMC to invest in the field. We have now a new cancer center director with a cancer immunologist. You have seen on the video, Dr. Ferris, who works also with Dr. Stanley Marks, who is the chairman of the UPMC Cancer Center. And I think cancer immunotherapy is really also a priority for the cancer center. A group of other talented investigators joined the group to increase the depth and breadth of research in the cancer immunology field. And as Chuck said, the institution, UPMC, is heavily involved in the field with this new immunotherapy center, not only for cancer, but also for other diseases, aging, and so on, and also a new cancer hospital, and we are really excited by this opportunity. So, why cancer immunotherapy? So why did we believe that stimulating immune response was important? So there is a number of evidence now that show that tumor cells, like a little bit virus infected cells, can stimulate immune response. We know that cancer cells can produce antigen, or present antigen, or release antigen in the tumor microenvironment. We know that some antigen-presenting cells can catch this antigen and stimulate a subset of white cells called the lymphocyte, the T lymphocyte, that expand in the lymph node, expand in the circulation, use some receptor to traffic back in the tumor microenvironment where they can encounter tumor and kill tumor. So these are the steps of the tumor immunity cycle. And what we have learned about this past 50 years is that all stages of this immune response, there are some good actors that enhance immune response. There are in green, and many of them, which can be different type of molecule or cytokine, and other one which have a deleterious effect, which impede this immune response. So as a cancer immunologist, what we want to do, and we thought that we could do that for years and years using cancer vaccine, we want to stimulate high level of this circulating T cell capable of recognizing the tumor, a little bit like we do for virus infection, right? Or when you get a vaccine for flu, in order to stimulate a high level of this lymphocyte that can kill the virus infected cell. We thought that we could do the same thing for cancer. So we have used a different type of vaccine approach to do that. We wanted to stimulate these high-level T cells of good quality, capable of recognizing well the tumor, cells that persist in the body of patient, and cells that could traffic back to the tumor site where they could encounter the tumor cells and kill the tumor cells. And we did all that with different type of vaccine, but this was not enough. So the patient had high level of these cells, but we had this paradoxical coexistence of immune response, but still no clinical benefit. I think one of the major breakthrough in the field of cancer immunotherapy has been to understand this last point, the mechanism used by tumor to fool the immune system, the so-called mechanism of tumor-induced immunosuppression. There are many that I would like to review briefly with you, and I think the big success of current immunotherapy, which are approved now for treatment of cancer, has been to counteract this immunosuppression. So if we review this mechanism, and I would call them immunoregulatory pathways, so this is a schematic presentation of the tumor microenvironment. This is, let's say, a lymphocyte, so a T cell capable of recognizing tumor cells. So they are activated by tumor antigen presented by the tumor, which is good. So there is immune response, but too much of a good thing is not always good, it's bad. When these cells get hyperactivated by too much antigen load, they got tired, they got so-called exhausted, and express a number of markers of exhaustion, the so-called inhibitory receptor or immune checkpoint receptor. And so this inhibitory receptor contribute to promote T cell dysfunction. They interact with their ligand in the tumor microenvironment and promote T cell dysfunction. There are also in the tumor microenvironment other components of the tumor microenvironment that promote this T cell dysfunction, a number of suppressive cells that suppress immune response. There is also a metabolic component in the tumor microenvironment. The tumor and the immune cells compete for nutrients, amino acid, oxygen, and the tumor wins the battle, leave little of oxygen and little nutrients to the immune cells that end up dying. The tumor also can produce a number of soluble mediators like IL-10 or other one, which impede the survival of the immune cell in the tumor microenvironment. So we need to find a way to counteract this mechanism. So, and based on this knowledge of immunology, we have designed a number of targeted immunotherapy to counteract this mechanism. The most famous one is what you may have heard, immune checkpoint blockade, and antibodies targeting this inhibitory receptor, CTLA-4. This is the antibody called ipilimumab, which is FDA approved in melanoma and also in lung. PD-1, which is another inhibitory receptor targeted by multiple types of antibodies. This is a big success of immunotherapy in the clinic, as I will talk to you a little bit later. But ongoing other immunotherapy, most part of the time in combination with immune checkpoint blockade, target the soluble mediator, targeting the suppressive cells, and also new approach to target the metabolism in the tumor microenvironment. So the breakthrough in the field of cancer immunotherapy was really this trial in melanoma, where patients with advanced melanoma were treated with this antibody targeting this inhibitor receptor, so-called immune checkpoint blockade, this anti-CTLA-4, this antibody which is now made by BMS called ipilimumab or Yervoy, targeting this receptor. And what we have seen for the first time is evidence of clinical response in 10 to 15% of the patients with advanced melanoma, which was a major breakthrough. It was a disease which was not cured previously. And most interestingly, not only there is this response, so shrinkage of tumor, but this prolonged effect on survival. So now we have almost 10 years follow-up on this trial, and there is about nearly 20 to 23% of the patients that are still alive after having received only 4 doses of the immunotherapy. So this is the beauty of cancer immunology, cancer immunotherapy. You stimulate immune response, you stimulate these T cells, and the so-called memory T cells. When you stimulate these cells, they keep them in your body, they are there, and if the tumor comes back, they are reactivated and are capable of providing anti-tumor effect. That's really the beauty of this type of treatment. So, but even better than CTLA-4, similar approach targeting another immune checkpoint blockade, a molecule expressed by lymphocyte called PD-1. This is an inhibitor receptor, but here in melanoma, it's not 10%. We're talking about 30 to 40% of patients with advanced melanoma exhibiting shrinkage of their tumor. So multiple antibodies are available now and are FDA approved, nivolumab or Opdivo, which is the antibody by BMS, or Pembro or Keytruda that are the most used and most known. And anti-PD-1 is not only approved, FDA approved for melanoma, but it has shown proof of efficacy in multiple other cancers, non-small cell lung cancer, kidney cancer, Hodgkin lymphoma, amazing effect for Hodgkin lymphoma, head and neck cancer, MSI-high cancer, hepatocellular carcinoma, and the list gets longer years after years. So anti-PD-1, so these are the trials that show the efficacy of anti-PD-1. This is a classical waterfall plot where you have the evaluation of tumor size in patients treated with anti-PD-1. So this patient didn't benefit, you have an increase in size, but here the majority of the patients should decrease in size, right? And 40% had a significant decrease in size, objective clinical response. And here again, if you look at the survival, you have this plateau here, right? And this long effect. Now we have at ASCO, I think very soon, we're going to present an update of this pembrolizumab trial, and at 5 years, you still have 30 to 40% of the patients that are still alive. So again, these memory T cells are in your body, are there, and can fight the tumor cells for a very long time, and this is really a big advantage of cancer immunotherapy. So with the efficacy came, of course, adverse events. You unleash immune response against tumor antigen and sometimes against normal antigen. So as I said, sometimes some major side effects, mainly with CTLA-4. Fortunately for PD-1, the effects are less frequent, 14% of grade 3 or 4 side effect, and most part of the time is fatigue. So, but there is a new now symptomatology that comes. You know, this patient develop autoimmune disease that you need to manage. Some rare autoimmune bullous dermatosis like bullous pemphigoid. Some people develop some vitiligo, which is more benign. And I think there is a lot of reason to believe that the occurrence of this adverse event can also predict response in most part of the patient. So now that cancer immunotherapy become standard of care and maybe has become one of the most potent therapy of cancer, there is a number of questions that come. The first is how to predict response. So I said 30 to 40% of the patient melanoma, the response still 2/3 do not, right? And in non-small cell lung cancer, it's like more like 88% of the patient do not respond. So is there a way to predict response, the responder? And this is a very active area of investigation. So both in academic center and in company, a number of markers have been proposed. None of them is perfect, but still they enrich. And if you have this marker, you are more likely to respond. This is a PD-L1 expression, which is a ligand that binds to PD-1. If the tumor expresses it, you are more likely to respond. And it's even a companion diagnosis for the treatment by pembrolizumab for non-small cell lung cancer. The mutation load, so if a tumor has more mutation, is more likely to present some neoepitopes, neoantigens that are more immunogenic and more likely to respond to immune checkpoint blockade. The presence of a spontaneous T cell in the tumor microenvironment, so indicating that the tumor is spontaneously immunogenic, makes you more likely to respond to immune checkpoint blockade. So most recently, Merck came also with a gene signature, an interferon gamma gene signature, which is another way to say that the tumor is spontaneously immunogenic. So the second question is the patient, the most part of the patients still do not respond. So there is a mechanism that support the non-response, what we call refractory, so and innate refractory or secondary escape to the treatment. And it's also an active area of investigation where we take the biopsy of the patient pre and post-treatment, use immunogenomics approach to try to find out what are the pathways that are involved in the resistance that may occur spontaneously or after initial response. Some patients may escape, so we try to find out what are these pathways. A few of them have been identified and in order to target them with additional treatment. And another important question is now that this immunotherapy have shown efficacy in the clinic, what can we do? So beyond PD-1 providing clinical benefit to 30 to 14% of patients with melanoma and other type of cancer, what can we do? And what strategy shall we adopt to decide what to do next? So that's basically what my lab is doing and other people are doing, and mostly looking at what other inhibitory pathways are really active in the tumor microenvironment that can be targeted with blocking monoclonal antibody. So this is what's happening all over the world. So a colleague of mine in biopharma takes some time to try to count all the treatment with immunotherapy, including in the trial, and he counted approximately 1,000 of clinical trials worldwide, including immunotherapy, and most part of the time is immune checkpoint blockade. And you can see that's what the biopharma are doing now, combining immune checkpoint blockade, anti-CTLA-4, anti-PD-1 with other more classical treatment of cancer, targeting this hallmark of cancer. Can be targeted therapy, VEGF inhibitor, radiation oncology, and so on. So many treatments are going on in order to further improve the efficacy of this immune checkpoint blockade. So another way to approach the development of immunotherapy in order to improve the benefit of current immunotherapy is to come back to what we know about this tumor immunity cycle, each step that leads to potent T-cell response and address, meaning the pitfalls. For example, I said that the tumor cells can present antigen, can produce antigen. Some do not do a great job in producing antigen and presenting antigen. So maybe what we want to do in this case is to use additive approach to induce tumor cell death, to promote release of tumor antigen. And this can be achieved by chemotherapy, radiation oncology, another way to achieve this goal, or targeted therapy. So if the problem is the antigen presentation, meaning the cells, antigen-presenting cells supposed to catch the antigen and present to the immune system are not very good, you can design other approach with specific antibody and anti-CD40 antibody or TLR agonist in order to improve their antigen capacity. If you don't have immune cells in the tumor microenvironment, you may be able to bring them, we call adoptive transfer of cells, which can be transferred with a specific T-cell receptor, also called the CAR T cells, right? And if the cells are there in the tumor microenvironment close to the tumor, but cannot do a great job because this inhibitory pathway are present. So beyond PD-1, there is a number of other pathways. And I think one very promising approach has been to combine anti-PD-1 with IDO inhibitor. I think we're going to hear about this combination at ASCO this year. So overall, to come back to the biology of cancer, with different histology, we recognize that there is mainly three main cancer immunophenotypes. An inflamed tumor, so a tumor which is immunogenic, capable of stimulating immune response. The immune cells are there, but still it's not enough to provide anti-tumor activity because there is a number of cells that have a deleterious effect on this immune response and pathways that are activated. That's why you need to come with immune checkpoint blockade or depleting the suppressive cells and so on to counteract this mechanism. Another phenotype is immune-excluded phenotype, and this is true, for example, for pancreatic tumor. What happens here, the tumor may be immunogenic, the T cells may be there, but they are kept at distance from the tumor because the so-called cancer-associated fibroblast protein produce a protein that make a coat around the tumor and don't let the T cells infiltrating the tumor microenvironment. In this case, so the investigators have identified some specific marker of cancer-associated fibroblasts that can be targeted with specific antibody to deplete them. And this is an ongoing trial from Bristol Myers, which really addresses this issue. And hopefully we're going to hear about this trial very soon. And another phenotype is, meaning the cold tumor, the non-inflamed tumor. The tumor don't present antigen. For some reason, don't stimulate immune response. So, and this also, a number of investigators have found that this may be due to the activation of a number of oncogenic pathways that prevent the tumor to be immunogenic. And we can target this oncogenic pathway with some small molecules. So this is also a number of ongoing trials are going to address this question. So I would like to showcase a number of things that is going on Pittsburgh, no specific trial based on our research from the bench to the bedside, and give you some example of new trial that we have promoted recently. The first one targeting the tumor microenvironment, the hypoxic tumor microenvironment with metformin. So metformin, as you know, is an anti-diabetic drug, and Greg Delgoff, who is a brilliant cancer immunologist at the Cancer Center, is evaluating this metabolic competition between tumor cells and T cells in the tumor microenvironment. They compete for nutrients, they compete for glucose, they compete for oxygen. And what he found is that metformin, this drug given to diabetic, has also the capability to reprogram the tumor cells. So it will decrease the consumption of oxygen by tumor cells, leaving more oxygen for the immune cells, allow them increasing the fitness of T cells, they will survive longer, do a better job, and has also shown that it acts synergistically with anti-PD-1, has published some very nice paper in mouse model showing when you combine anti-PD-1 with metformin, T cell function much better. And based on this data, we have initiated this month a first-in-human trial combining metformin and pembrolizumab for patients with advanced melanoma, hoping that this combination is going to do better than anti-PD-1 pembrolizumab alone, led by Dr. Najjar, who is a junior oncologist, and Dr. Kirkwood in Pittsburgh. So another example is the combination of anti-PD-1, pembrolizumab with PEG interferon alpha, and the idea here is to target the so-called immune desert. I said some tumor cannot be spontaneously immunogenic. A number of studies have shown that this due to activation of a oncogenic pathway or to the lack of interferon alpha, type 1 interferon pathway in the tumor microenvironment. And we thought that maybe a good way to counteract that is just to bring interferon alpha. Pegylated interferon alpha is available by Merck, so we wanted to evaluate this combination in patients with advanced melanoma treated with this combination. So it was a trial that I have included 43 patients. We have reached full accrual, and the response is promising. It's a small trial, but still nearly 50% of the patients have benefit. This will need to be confirmed in a large phase 2 trial, but it seems that we are here capable to doing a little bit better than pembrolizumab alone. Third approach that we are really pushing forward, and that is the work of my lab, is to try to combine blocking PD-1 with blocking other inhibitory pathway that may have a synergistic effect in combination with PD-1 blockade. And one pathway of interest called TIGIT. TIGIT is another inhibitory receptor like CTLA4 and PD-1, is highly expressed by T cells in the tumor microenvironment as often as PD-1. The ligand expressed by tumor cells are also highly expressed, and this interaction limits T cell survival. So we have evaluated in combination with our colleague at Bristol Myers, who produce the antibody for us, the immunogenicity of this dual blockade. We take tumor antigen-specific T cell from patient, and in an assay in vitro, when we do dual blockade, we do a better job than single blockade in increasing the function of these cells. We also show in mouse model that in purple here, when you do dual blockade in mouse-bearing tumor, this is a colon carcinoma, dual blockade does a better job of single blockade in prolonging the survival of the mice. So based on this data, we have designed a trial in combination with Bristol Myers. It's open in multiple centers worldwide, where we tried this combination, anti-TIGIT and anti-PD-1, in patients who do not respond to PD-1. And hopefully, we're going to convert PD-1 refractory patient in PD-1 responsive patient. So finally, I would like to showcase maybe another area of interest, and a very new area of interest is the role of the gut commensal, the gut microbiome, in modulating immune response to immune checkpoint blockade. And these data come from mouse data that show that some mice with tumor are not responsive to immune checkpoint blockade, but can be rendered responsive to immune checkpoint blockade if you give them a specific bacterial commensal, or even fecal microbiota transplant from a responder mice, and you can convert the non-responsive mice in a responsive mice. So what we and others did is to try to see if the same thing was true in patients. What we did is get the stool from a number of patients treated with a PD-1 inhibitor, and found that the responder patient, as opposed to non-responder, are enriched, the stool of the responder are enriched in the number of commensal, bacterial commensal. And based on this data, I think the end game for us will be to identify what are these commensals and hopefully design some type of prebiotics that you can give to patients in addition to PD-1. But I think we need more work to really find, all the gut commensal profile linked to this response. This is an ongoing work, as I said. So, but as we wanted to move forward and to validate this concept, we can we use a gut microbiome to render a patient responsive to PD-1. We have designed a trial where patients are going to be treated with pembrolizumab, which is standard of care, right? After, usually you evaluate the patient after at 12 weeks, after 3 months, you have 1/3 of the patient are supposed to respond, 2/3 will not respond. These are melanoma patients, and we're going to treat the non-responder patient with one single administration of fecal transplant from a long-term responder. So as you may know, fecal transplant is commonly used to treat infection with Clostridium difficile. It's pretty safe. And we have now developed a fecal transplant lab in the Cancer Institute who is going to do the trial. So the first patient is going to be included in March. And so we are very excited by this trial. We're going to learn more about the role of the microbiome in modulating response to cancer and to immune checkpoint blockade. So this slide is just to tell you the main challenge. So out of the fact that we need to have an active research program to find new ways, new approaches to improve clinical benefit to patients. So when we have a potential pathway, so my lab has been working on 2 inhibitory pathway, TIM3, I talked to you about TIGIT. We show evidence that blocking TIM3 in addition to PD-1 was more efficacious than PD-1 blockade alone. It was 2010. We had to wait 5 years for the first trial picked by Novartis and then another company, Tesaro. So TIGIT, the inhibitor receptor, we also demonstrate that dual blockade PD-1 TIGIT, and I show you some of the data, did a better job than PD-1 blockade alone. We report the data very close to our colleague at Genentech, 2014-15, a year after the trial already was implemented in patients. So I think the biopharma now are putting a lot of resources in this trial. The field is moving fast, and the challenge for us is to be able to follow the pace, be able to open this trial very quick and to include the patient also at a great pace. So I'm going to end up my talk here and hope that I convince you that cancer immunotherapy was important, the standard of care for multiple cancer, and there is room for improvement, and we have some clue about what to do next to move our research funding in collaboration with academic center and biopharma into the clinic with the help of a multiple funding agency, and it's good to have ITTC and University of Pittsburgh pitching in to help us to reach this goal, to give opportunity for cancer patients, that's our goal, right, and to fill this unmet need using this first-in-human clinical trial within the UPMC Cancer Network and with also multi-center worldwide, and also take the opportunity with our cross-disciplinary research to learn from this trial to generate the next generation of immunotherapy and training the next generation of physician scientists. I would like to end my talk and thank you for your attention.
So thank you very much indeed, Dr. Zarour. We have time if there are any questions for Dr. Zarour in relation to the immunotherapy of cancer.
Thanks. Is that on? Thanks. That was a great talk, and particularly enjoyed the innovative stuff you're doing with microbiome transplants and metformin in combination with the immunotherapies, which we're all using already. And it's hopefully we'll be doing some of that stuff in Cork in the not too distant future. Are there any stimulatory molecules which you want to, rather than inhibit pathways, you can actually stimulate exhausted immune cells to get back to work?
Yeah, so this is also an approach, right? I talk mainly about relieving the brake, right? And a number of companies now not only are relieving the brake with PD-1 and CTLA-4, but are pushing on the accelerator by activating this pathway. So I think BMS has a very advanced trial using 4-1BB in combination with PD-1 blockade. I haven't heard recently about the results of that, right? Some company, I heard also X40 go along the same way, although the data doesn't seem to be very good for X40, right? That's, so I hope that some data are going to be presented at ASCO. I haven't heard until now major success of such an approach.